Hello, everyone, and welcome to today's webinar titled New Tools to Tackle Multiplex Imaging Challenges in Spatial Proteomics. This webinar is a part of the Immuno-Oncology virtual event series. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by Thermo Fisher Scientific. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you happen to have any trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits link located within the abstract on the left of your screen and follow the process to obtain your credits. Okay, now let's get started. I now present today's speakers, Chris Langsdorf, Product Manager at Thermo Fisher Scientific, and Adyary Fallarero, Senior Product Manager, Thermo Fisher Scientific. For a complete biography on our speakers, please find it to the left of your screen. Chris and Adyary, you may now begin your presentation.
Good morning or good evening, everyone, depending on where you are in the world. My name is Adyary Fallarero, and I am a Senior Manager in Product Management for Imaging Technologies at Thermo Fisher Scientific. I'm located in Finland.
Hello, everyone. My name is Chris Langsdorf, and I'm a Global Product Manager in Protein and Cell Analysis, also at Thermo Fisher Scientific, and I'm in the United States.
We are both super grateful to have you all attending our talk. And today we will be sharing with you all of the new tools that our company has recently developed to advance multiplex imaging for spatial proteomics research.
We hope you'll benefit and gain new knowledge on spatial biology. Feel free to get in contact with us and our teams afterwards if you need any more information or if you have questions. So let's get started. So we'll jump right in and look at new tools to tackle multiplex imaging challenges in spatial proteomics. So our agenda today, we'll look briefly at the revolution in spatial biology, and first look at some challenges that pop up when we start to look at multiplex proteomic imaging, and then look at how we've addressed these challenges, both using sample labeling and then also imaging and analysis techniques. So spatial biology really provides us better insights into our samples, primarily because we're able to analyze not just single cells, but look at their context in a larger environment. So we can use antibody-based detection to tell us what cell types are present, but also the state of those cells, so their metabolic or activation state, for example, and what functions they're carrying out. Because our samples are generally fixed, we can get an idea of their identity in both time and in space. And by studying multiple cell types simultaneously, we can understand the interaction of those cells and how they form larger and more complex neighborhoods or networks. And so we can begin to understand both the tissue microenvironment and the architecture of our sample in its native state. So let's look at an example of how this works. We can image an entire tissue section, and after imaging this, we can zoom in on one section of the sample and see how we can understand the spatial context in the sample. So we can see, as displayed with multiple different colors, we can see the location of different cell types as they exist in their native environment. Then we can use our analysis tools to mask and identify the different cell types that we've used antibody-based detection to label. And we can go a step further with our analysis and ask questions like, what is the nearest neighbor to each of these cell types? And begin to understand the network and the architecture of our sample in much greater detail. Now, several challenges pop up when we switch to this type of spatial workflow. Generally with microscopy, we use primary antibodies followed by fluorescently labeled secondaries. When we move from 3 or 4 antibody-based targets up to 8 in this example, secondary antibodies are no longer useful. We need to pivot and begin using primary antibody conjugates. And as we see here, the primary antibody labeling workflow is very fast. We can make a cocktail of all of these antibodies and label our sample in about an hour. There are some drawbacks, though, is that there will generally be a limited catalog menu available of these conjugates. And unlike with secondary-based detection of 3 or 4 targets, where we always choose the 3 or 4 brightest dyes available, here we need to choose dyes based on their spectral properties and not necessarily on their brightness. So fortunately, there's a solution to this challenge, and this is provided by the Alora spatial amplification reagents. So here we use a 3-step workflow. First, we label our sample with a primary antibody, followed by a wash, and then labeling with a secondary antibody HRP conjugate. After this, we add our Alora spatial amplification reagents, which are covalently attached to our sample in the location of our antibody binding. After this step, all of the antibodies are stripped off our sample, leaving just a very bright fluorescent labeling in the location of each antigen that was originally present. And so, as we can see on the left, after one round of labeling, we can see the location of one antigen of interest. After repeating, we can see a second, a third, and in fact up to 8 plex with great sensitivity, so very bright signal. And this gives us a lot of flexibility in how we choose and how we use our antibodies. Because amplification is provided, we can use substantially less antibody, and we're not required to covalently attach a dye to our antibody. The drawback here is it's a longer workflow, so we have to go through multiple labeling cycles. Now, the Alora spatial amplification reagents are compatible with primary antibody conjugates. So you see 2 examples here where the primary antibody conjugates are labeled with a few of our brightest dyes available. So Alexa Fluor 647, 594 give us great signal to background for these primary conjugates. But with either a less bright dye or a lower expressed antigen, we switch to the Alora spatial amplification reagents to give us that signal to background that we need. As far as availability of primary antibody conjugates, there are a few ways besides just acquiring these from a catalog. These are quite available to produce as a DIY method. So there are kits available that make it very easy to label your antibody with your dye of choice that's compatible with the system, either by assembling a kit using your own purification system, or we have a technology called Ready Label that does 2 things. It both purifies your antibody and then labels it with one of these 8 compatible dyes and gives you a purified conjugate. Or if you like, ship your antibody to us. Our experts will conjugate it and return it back to you. So there's a lot of flexibility and convenience available with these options. However, they can be a bit more expensive than just acquiring a conjugated antibody from a catalog.
Well, thanks so much to Chris for sharing with us the labeling tools that we've been developing to advance spatial proteomics research. Now we're going to switch gear a bit and focus on instrumentation, so fluorescent microscopy instrumentation. You all are very aware that fluorescent microscopy can be and has been extensively used to investigate how single cells are organized into a tissue microenvironment, and none of the currently available technologies are really perfect. They all have benefits and drawbacks. So what we've tried to do from our perspective is that we have tried to tackle some of the drawbacks of the currently available instrumentations and try to progress in that manner the type of research that spatial biology scientists can perform. I start maybe with a bit of an overview of what we currently have available and then how we have addressed the current limitations of the existing technologies. I think you are all very well aware that a great body of research that has been done around spatial proteomics has been done using cyclic labeling and imaging. Imaging that is done in this approach is really pretty conventional. So there is between 2 to 4 fluorescent channels that are used on those instruments. And the benefit of this approach is that because it is performed in a cyclic manner, well, this allows for a high number of targets, protein targets that can be interrogated. The drawback, on the other hand, is that because it's a cyclic process, it tends to be of a lower throughput, and also because it is constant illumination of the sample and they go through certain rounds of stripping, then obviously that can damage the integrity of the tissue. The approach that we have taken and that we have tried to address the current limitations, it's really spectral imaging. And spectral imaging allowed for the capture of simultaneous fluorophores within one single round. So instead of only doing the conventional 2 to 4 channels, maybe even 5 fluorescent channels in one single go, so the aim of a spectral imaging is to do even more than that. So to capture between 7 to 10, even sometimes more fluorophores in a single round. Obviously, there is an advantage from doing this, and it is the fact that tissue gets better preserved. But obviously, there is also a drawback. And the drawback comes from the fact that when there is so many fluorophores that gets acquired in a single imaging round, there is obviously signal bleed through that needs to be eliminated. So if we try to understand a bit more, why is this drawback existing in spectral imaging and where is this coming from, then we can go to the root cause of that problem. And the root cause is really that all the fluorophores that I use, or the majority of the fluorophores that I use for spatial imaging, they do have a very broad emission spectrum. And if you can see in this example, it's a kind of quite clear example that if we are, let's say, imaging ECAD in a 514 channel, the emission spectra of the 514 channel will also appear and show up in both neighboring channels, so in 488 and in 555. So in order to be able to resolve that signal and to remove that bleed through, we can apply a mathematical processing that is called unmixing. And unmixing is not really something extraordinary. It's a very relatively straightforward process by which we can establish the relative contribution that each fluorophore that is used on that plex, on that specific protocol, we can establish the relative contribution from each of these fluorophore to every pixel of the image. And that's done in a sort of relatively simple way by acquiring spectral signatures from the unstained tissue. So just to account for the typical autofluorescence that you see in biological samples, and then from the individually stained fluorophores on the same tissue where the assay is being performed. Using spectral unmixing, then protein targets can be easily resolved and discriminated even if the fluorescent spectral signals overlap. And as you can see on the bottom images that are shown here, ECAD in the 514 channel can then very clearly discriminated from the CD163 and the PD1 signal that appears on the two neighboring channels. So this main, let's say, drawback associated with spectral technologies has been something that we truly has wanted to address with our new instrument. Our new instrument is just launching now in this fall, and it's called Invitrogen EVOS S1000 Spatial Imaging System. we have paid a lot of attention on simplifying the mixing workflow, keeping it very, very straightforward, and it also performed in a way that it is part of the acquisition. So it's done on the fly while the images are being acquired. And it's not only that, it's also that we've gone one step forward and we have also tried to verify the quality of the unmixing. So it's not only about looking at the images and trying to figure out whether they look okay, we are also providing substantive data as part of a quality report that allows the user to understand the quality of the unmixing that's been performed. But we have gone beyond that. So it's not only about improving the mixing workflows, but also about addressing other challenges that are encountered with currently existing spectral imagers. One of them is the fact that they are often restricted to very specific labeling methods, and we've tried to expand on those, and you will see just shortly after this that we've tried to expand the number of dyes that can be utilized and the type of technologies that can be utilized with EVOS S1000. We've also tried to do it in a fast way, so you don't have to spend hours to acquire a 9-plex. You can do it in a single round. EVOS S1000 would allow the users to acquire a protein target and DAPI, so that makes for the 9-plex, and also keeping a high resolution. So the resolution that we currently have, it's 325 nanometers per pixel at 20x. And last but not least, it's all about making spatial proteomics and spatial biology in general accessible to a broader audience so that many more researchers can utilize spatial biology and can study biological processes using these tools. So we are not making it simple. That would be kind of something that would be not correct to say, but we are trying to make it simpler. So let's go now through all of those challenges that I was initially addressing that are presented in currently existing spectral imagers. So the first one is really that unmixing tends to be limited to very, very specific dyes that are very often proprietary dyes. So we've tried to really address that issue. When you generate the protocol, when you create a protocol with EVOS S1000, I'm showing you here a video of how that process looks like. You will be presented with a repertoire of dyes. It's roughly around 30 dyes. That includes Alexa Fluor, Alexa Fluor Plus, and also a spatial amplification reagents such as Alluras and Opals dyes. You can select the dyes. In this case, we have selected Alexa Fluor, so we can select all of the Alexa Fluors that are existing, and then we can generate a protocol that will immediately generate a channel configuration suitable for that 9-plex that has been defined. You see that the software will show you the overlap that exists on the emission spectra of those dyes that has been selected. And what we do is that then we set up a channel configuration automatically that will acquire the primary channels, so those are the ones shown in blue, as well as the support channels or secondary channels. Those are the ones that are needed in order for us to eliminate this bleed through that comes from the proximity of the spectral signals. But that is something that is done completely automatically and that it doesn't need to be set up with any user input. The user, of course, can also define the protein targets that are on the assay. So if you have, for example, nuclei, if you have SMA, whichever targets are defined. And then once more, I want to highlight that it's a setting. The protocol is set up to be compatible not only with primary conjugates, but also with spatial amplification reagents, so that there is flexibility to use any kind of labeling solution. Another challenge that we have tried to address, and I have to say that this is probably the core challenge that it's impacts more when scientists want to go beyond a 7-plex or 6 protein targets and DAPI, is that it becomes really troublesome. It becomes really complicated how to perform unmixing for anything higher than a 7-plex. Our solution has been that first and foremost, we are using the same unmixing workflow regardless of the plex. So if you go to a 7-plex, it's the same protocol or it's the same workflow if you all the way to a 9-plex. The idea is that first, an unmixing matrix needs to be generated, so we need to extract spectral data from images. So users will need to first acquire an image of the unstained tissue slide, and then individual single color calibrator slides that needs to be also generated as part of the initial setting of the unmixing matrix. So once that happens, you can then utilize that protocol, and then you can start setting up an assay, a 9-plex. So your protocol will be loaded with all of the 9-plex, and you will first, of course, set up the illumination parameters in all of the channels. The first example that I'm showing you here is DAPI, so we can easily autofocus and find where the tissue is with DAPI. Then we can check all of the channels. For simplicity, I'm only showing here DAPI. And then once you do that, you can set your area scan. And the area scan can have, of course, multiple dimensions. You can capture the whole slide if you so wish. In this example, we are setting a 2 square millimeters area scan. And once that has been set up, then of course the images will start to be acquired, and the images can be acquired then now entirely automatically. So the system will start to acquire images in every single field of view, all of the primary and support channels that were defined in the channel configuration. And also at the same time as those images are generated, so right after a field of view is completed, then Unmixing will be triggered, and it will kind of be part of the image acquisition workflow. So at the end, you can acquire a 1 square centimeter 9-plex image that is fully unmixed, that is fully stitched, and it's also reported in an OMET file format, so completely standardized file format. And that can happen roughly within 1 hour using a magnification of 20x. You see now how the image, this is in real time, so the actual acquisition done by the instrument. Now the image is generated. What you're looking at now is the image that has been generated and that it's already unmixed. From that point on, you can auto contrast the image. You can then toggle on and off multiple channels. So now I'm giving you some examples of some of the channels being toggled on and off. You can go a bit deeper and zoom in into a certain structure that you would be interested. And now you can compare a certain channel, the raw signal, this is 514, with the unmixed signal. So you can see exactly how this signal bleed through that we were discussing initially in the channel configuration, how that can definitely be removed as part of the unmixing program, the unmixing workflow. There is definitely then no need for any post-processing after this unmixing has been generated. So very often what we see is that users cannot assess only subjectively, or should not only subjectively assess the quality of the unmixing, and that's an issue that we have tried to address. So once the unmixing metric is generated with the EVOS S1000, you will be prompted to if you want to also generate an unmixing quality metrics report, and that's exactly what you see now in the video that it's happening. So we generate an unmixing quality metrics reports that do not only contain the raw images versus the unmixed image, so you can do a kind of a qualitative assessment of the results, but we also generate quantitative parameters that allow the user to very easily estimate how the quality of the unmixing has gone. We provide very clear cutoff values from those parameters so that the user can assess. We are also providing troubleshooting tips in case that there's been some issues that could be addressed, and the user can then go back and repeat or reassess the process that has been done so that those parameters can be improved. In our view, this is something very positive that truly improved the transparency and really addresses the fact that it doesn't need to be a black box approach to unmixing. And of course, last but not least, I think that every system and every way to do microscopy needs to be sufficiently simple and sufficiently intuitive, even for users that are not used to the space, even for users that are new to the space. And here I'm just showing you some of the ways in which you can interact with the instrument and explore your tissue. So we have a proprietary feature that we call periscope mode. So you can turn on any channel and then explore your tissue and navigate the tissue to investigate what are the expression of the targets and how the tissue landscape actually looks like. This is something that becomes very, very practical and very simple for even new users to interact with. And we do truly believe that we are providing a sophisticated simplicity that will allow anyone entering the space to be able to quickly gain confidence and easily set up, acquire, and visualize 9-plex images. But I was referring to you of the fact that EVOS S1000 can be openly and flexibly utilized with multiple different kinds of labeling strategies. So here I am showing you some examples of the results that can be obtained when using Allura special amplification reagents. This is a very effective labeling strategy for targets that have very low expression levels. It does require multiple iterations and multiple cycles of staining, but the result is like a very bright signal that can be very easily discriminated between targets that even have low expression levels. On the other hand, you can also utilize the EVOS S1000 with primary conjugates, and by using primary conjugates, you can set up a very quickly labeling step. It can go as quickly as 1 hour with our own validated primary conjugates, and then the labeling and the capturing can be completed within a very short period of time. So if we do, for example, a 9-plex with primary conjugates, as such that we are offering within Thermo Fisher, but it can also be any other primary conjugate, then you can do the labeling and then capture the images all within about 2 hours at 20x. And that would include, again, the unmixing and all the stitching and processing of the data to generate an OEMITIF file. If you do have primary antibodies that for which the conjugates are not currently available, then another possibility is to directly conjugate those primary antibodies that you are used to utilize. And that is through the use of these ready label kits that Chris was introducing in the first part of our talk. This is the kind of results that you can obtain. This is a 9-plex composite, and you can see that it's very relatively easy to do on your own, and it would also be truly compatible with the use of the EVOS S1000. So I hope that I have been able to convey the idea that the EVOS S1000 spatial imaging system, it truly is a new novel alternative in the spatial imaging space. We do hope to provide a choice of achieving high multiplexability in a single imaging step and not having to deal with the complexity of a very laborious unmixing workflow. We have tried to make it simple. We have tried to make it also in a manner that provides you confidence, that has quality metrics that can be reliable to your results and to your approach, and also give you flexibility. So you can utilize spatial amplification reagents, such as our Allura dyes, but you can also use validated primary antibody conjugates, or you can generate your own conjugates using the ready label kits that they were just mentioned. We are happy to answer any questions that you might have, and we are very happy to also be one of the partners that can help you in your spatial biology journey. Thank you so much for joining.
All right. Thank you both for your very informative presentation. We will now jump into the Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. Okay, let's go ahead and get started. Okay, the first question that we have here says, how have you made easier spectral unmixing?
Maybe I can take that one, Chris. That's okay for you? So spatial biology, as many of you probably already have learned, it's not easy. It's quite difficult, and it can be quite daunting. And if you are approaching spatial biology, and particularly spatial proteomics, using spectral imaging, then you know that a critical component of spectral imaging is unmixing, and unmixing can be very, very difficult when you first start understanding how it's even done. So this is one of the things that we try to address with the EVOS S1000. So we try to make it simpler. It's inherently complex, so we cannot make it simple, but simpler. And we start by, first of all, offering default protocols. So there are protocols that can be ready to use that already contain unmixing metrics that can be utilized in an assay. Those are default protocols that have been generated on tissue that are like kind of reference tissue. So, and they have like certain fluorophore combinations. So those you can start using right away. What we do recommend is that once you start using the instruments more frequently, that then you generate your own unmixing metrics or your own protocol that contains an unmixing metrics. And for that, we offer very clear instructions, like a guided workflow on how those single color control images can be obtained. And we do think that that's the best approach. It will give you a very precise extraction of the spectra. It will generate very clear discrimination of the signal. So your 9-plex will have a really, really high quality. There is another component on doing it from scratch and doing it on your own, and it's that every time that you generate the protocol and you go through the single color calibrator workflow, then there will be also an unmixing quality metrics report that gets to be created. And that unmixing quality metrics report, it's fantastic from the standpoint that you can see what the expected result is going to be even before you start performing area scanning. There are qualitative and quantitative parameters on the report that you can very quickly inspect that can give you like a very quick overview that, okay, this is going to work. And then once you have made a decision that, okay, things are looking okay, there's no need to do any changes, then you can start scanning. And that then gives you the biggest benefit. The scanning from that point on has on-the-fly unmixing. You don't have to worry about doing any post-processing. Every time that you scan a 9-plex, as you saw in the video that we have just seen on the instrument, is actually already happening on mixing on the flight. And it doesn't matter if you're doing a 7-plex or an 8-plex or a 9-plex, the image will be generated fully on mix and stitch, ready to use. So it really is facilitating the work that you might do then afterwards. So that's how we have tried to make it as simple as possible.
Perfect. Thanks, Adyary. Okay, the next question that we have here says, can the labeling solutions you have presented be combined?
I can take that one. So yeah, great question. And really all the different labeling solutions that we discussed today were designed to be combined in a seamless way. So that all the colors are perfectly aligned with the channels on the EVOS S1000. And then really the thing to consider is what order you would apply these to your sample. So generally, if you need amplification of a target, we'll use the Alora spatial amplification reagents first, either one or multiple of those sequentially. And then following that, we'll label with our primary antibody conjugates. And then whether those primary antibody conjugates are direct from a catalog or you make these yourself using a labeling kit, either way is fine, but then we would generally apply those as the second part of that experimental protocol. So great question, and yep, definitely everything is designed to be seamless and work together to label your tissue, just depending on what targets you're hoping to study. So thank you.
Perfect. Okay, question 3 here. Can any dye be imaged with the EVOS S1000?
That's a really good question, and one question that we get very often. So hardware-wise, the instrument can detect more than 60 plus configurations, about 65 configurations. So meaning that we can detect very many combinations of excitation and emission, which means that pretty much anything that is in the visible spectra we can detect. In order to keep it very simple, we have a preset list of fluorophores that are already in the software, so that the user just need to select the fluorophore. And those fluorophores include the majority of those that are kind of very widely used in spatial biology. So Alexa fluors, Alexa fluor plus. We have also E-fluors and the special amplification, so pyramid amplification labels, such as the Allura special amplification reagents and opals as well. So you just have to pick the fluorophore, and then the correct channel combination will be selected, and then you can build your protocol, whichever combination of fluorophores. We will educate the user if the combination is not correct. So it won't allow you to make complications that are kind of not possible. And it will also, when the combination is selected, it will also, unmixing will only be triggered when it's needed. So if the if the fluorophore selection is such that Unmixing is not required, then the software will also kind of automatically toggle Unmixing only if it is needed. So the short answer is that we can pretty much image anything. It can be a spatial amplification as well as primary conjugates. The list of fluorophores currently is about 35 fluorophores that we have. But of course, if you have spectrally compatible fluorophores, those can also be selected. And then as long as the user can set up the protocol and generate the Unmixing metrics, then the system would work. We are in the future, of course, looking to expand the list of fluorophores, so to make it even more flexible. But already at the moment, then you can use pretty much anything that it's available up there for spatial biology labeling.
Perfect. Thank you so much. Okay, it looks like we have one final question here. This one says, how many dyes can you image simultaneously?
Do you want to take that one, Chris?
Sure, I can take that one. Yeah, so the S1000 is really designed for simultaneous imaging of 9 colors, where 8 of those are to interrogate your antigen or protein of interest, and then the 9th channel is generally filled by DAPI for your nuclear counterstain.
And maybe just to add briefly that we have had some users that also have kind of used the instrument in a more cyclic manner, so that they just tick over sleep and then they can rescan the slide. It's not perhaps the most common scenario, but it's also something that we can support with the instrument.
Perfect. Okay, well, thank you again, Chris and Adyary, for your time today and for your very important research. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions we did not have time for and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 30th, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everybody, goodbye.
Thank you.